Ever find yourself just like completely in awe of, the human brain? It's constantly going, processing info, like orchestrating everything we do.
It's wild when you think about it, right? Like all our thoughts and feelings, all our actions, it all boils down to communication between these tiny little cells,
And
neurons.
that communication, that's what we're diving into today, synaptic transmission.
Mm.
We got this neuroscience textbook, neuroscience, exploring the brain. It's like getting a peek into how our brains work.
People compare the brain to a computer, but honestly, it's so much more intricate. Yeah, electrical signals are involved, but there's this whole chemical world going on that makes the brain truly amazing.
So it's not just electrical signals bouncing around like in those computers.
Not quite. We do have electrical synapses relying on electrical currents, and they are insanely fast, but they're less common, especially in our human brains.
The book mentioned crayfish. They have these crazy fast escape reflexes, and it's because of these electrical synapses, right?
Exactly. When speed is super important, like life or death situations, electrical synapses are key. Like almost instantaneous communication gives the crayfish that split-second advantage to, well, not get eaten.
Makes sense. But if we don't use those electrical synapses as much, what's the main way our neurons talk to each other?
Chemical synapses, they're everywhere in the human brain, trillions of connections, way more complex than electrical synapses could ever be.
Chemical synapses.
Huh.
So we're talking actual chemicals going between neurons, like tiny little messengers with instructions.
That's a great way to think about it. And you know who really started to uncover how these chemical synapses work? A scientist named Otto Lowy. And get this, his big breakthrough came to him in a dream.
Hold up. He figured out a major scientific discovery in a dream. Okay, I need to hear this.
So Lowy was studying how nerves control the heart, right?
Yeah.
And one night he wakes up from this dream with this incredibly vivid idea.
Okay.
He thought, what if he stimulated a frog's heart, then took the fluid from around that heart and put it on another frog's heart? His thinking was, if it's chemicals doing the talking between the nerves and the heart, he could transfer that effect.
Wow, that's some next level thinking right there, especially for a dream. So did it work?
It did. He called the mysterious substance Vegastoph, meaning vagus substance, since he was working with the vagus nerve.
Vagastoph.
Uh-huh.
It does sound kind of mysterious. But we've got a different name for it now, right?
We do. Today, we call it acetylcholine. And it does way more than just mess with the heart. It's essential for our muscles to move, showing how one single neurotransmitter can do a whole bunch of different things.
Acetylcholine, multitasking messenger. In our hearts, our muscles, it really hits you how much is packed into these little interactions constantly going on in our brains.
And we're just getting started. Lowy's discovery, it opened up this whole understanding of how the brain works. It paved the way for like decades of research into these fascinating neurotransmitters and how they do their thing. But to really get it, we need to zoom in even closer, look at the chemical synapse itself.
Okay, so take me to the chemical synapse. We've got acetylcholine, our chemical messenger. But how does it actually get from point A to point B, one neuron to the next?
Imagine you've shrunk down, like way down to the size of a molecule, right? And you're traveling through this huge landscape of the brain.
Okay, I'm picturing it.
You see these two neurons, close but not quite touching. There's this tiny gap between them, and that's the synaptic cleft. That's where all the action happens, where that chemical communication takes place.
So the synaptic cleft, that's like the main communication highway with messages constantly going back and forth.
That's it. Now, let's look at the neuron sending the message, the presynaptic neuron. At its end, it forms this structure, the presynaptic terminal. Think of it like a mailroom, packed with tiny packages all ready to be shipped out.
Okay, so like a mailroom, I'm with you.
Those packages, those are synaptic vesicles, and they're filled with thousands, just thousands of those neurotransmitter molecules.
So we've got our delivery trucks all loaded up and ready to go, and the synaptic cleft is the highway. Where do those delivery trucks go?
On the other side of that cleft is the postsynaptic neuron waiting for its delivery. It's got these receptors embedded in its membrane, like specialized mailboxes. Each one is designed to bind to a specific type of neurotransmitter.
Ah, so acetylcholine has its own designated mailbox.
You got it. It's all about that perfect fit, like a lock and key. The shape, the charge, everything has to line up perfectly for that neurotransmitter to bind to the receptor and actually deliver its message.
Amazing how something so tiny can be so picky. Well, we talked about different types of synapses before, right? Axodendritic, axosomatic, axoaxonic. Are they all using the same process to release neurotransmitters?
The process is the same, but where that synapse is located on the receiving neuron, that can change how much of an impact that signal has. The most common type, axidendritic synapses, that's where the axon of one neuron connects to the dendrite of another.
Okay, so that's the standard setup.
Right. But sometimes you get an axon connecting directly to the cell body, the soma. That's an axosomatic synapse.
Sounds like a more direct line of communication going straight to the cell body.
It is more direct, and it can have a much stronger effect on the receiving neuron's activity. And then there's the axoaxonic synapse.
Axoaxonic. That one sounds complicated.
It's where you have an axon terminal connecting to another axon terminal.
So instead of delivering the message directly, it's more like rerouting the message.
It's like fine-tuning the message before it even gets there. They can either boost or block the release of neurotransmitters from that other axon.
So it's like a volume knob for the message, adjusting it before it gets sent out.
Perfect analogy. And even within these different types, you have more variation. Some have larger active zones, which are the sites where those neurotransmitter vesicles release their contents.
A larger active zone, does that mean a stronger signal then? Like more delivery trucks at the same spot?
Exactly. It's another layer of complexity, showing how our brains can fine-tune their communication for, well, everything they do. And remember, we're still just talking about the structure of the synapse itself.
It's already so complex, and we're just scratching the surface. This is blowing my mind.
It gets even more intricate when we start looking at all the different neurotransmitters, what they do, how they affect us. Each one binds to its specific receptor, and it sets off this chain reaction that ultimately influences how that receiving neuron behaves. And that's where we really start to see how all of this intricate dance of chemicals leads to everything we think, feel, and do.
All right, we've explored this super intricate world of the chemical synapse, but now I really want to dig into these neurotransmitters. It's like we've been talking about the delivery system, but now I'm curious about the actual packages, what's inside.
And just like you can send a package in so many different ways, there's a ton of variety in these chemical messengers. The textbook breaks them down into three main categories: amino acids, amines, and peptides.
Amino acids, yeah, I've heard of those. They're like the building blocks of proteins, right?
Exactly, and some of them have this whole other job acting as neurotransmitters in the brain, like glutamate, for example. This one's the most common excitatory neurotransmitter, basically like the brain's accelerator pedal.
So when glutamate binds to its receptor, it's like hitting the gas.
Pretty much. It makes that receiving neuron more likely to fire, passing the message along.
Okay, I'm getting the picture. So if glutamate's hitting the gas, what's tapping the brakes?
That's where GABA comes in. It's the main inhibitory neurotransmitter, so it kind of puts on the brakes. And that balance between excitation and inhibition, that's key. Regulating mood, processing information, everything.
A constant balancing act. What about glycine? That's an amino acid neurotransmitter too, right?
Yep, glycine mainly works in the spinal cord and brainstem. It's involved in things like motor control and sensory processing. And like GABA, it's also inhibitory.
Gotcha. So we've got our fast-acting amino acids, but you also mentioned amines. What are those?
Amines, they're another group of these small molecule neurotransmitters, and they're involved in a whole bunch of things. We already talked about acetylcholine, which is crucial for muscle movement and heart rate, remember?
Right. It's coming back to me now.
And then there's dopamine, which often gets linked to reward and motivation, but it's actually more complex than just feeling good.
Dopamine. That's the one that's also involved in addiction, right? How does that work?
So dopamine is a major player in the brain's reward pathway. We do something pleasurable, dopamine gets released, and it kind of reinforces that behavior. Problem is, drugs can really mess with this system. They flood the brain with dopamine, and that's what leads to those intense cravings.
The reward system goes haywire. Fascinating.
Yeah.
A little scary.
Yeah.
Okay, so we've got acetylcholine, dopamine. There are more amines, right?
There are. Serotonin is another big one, often linked to mood, sleep, appetite, all that good stuff. That's why a lot of antidepressants target serotonin, trying to help regulate those emotional states. And then there's norepinephrine.
Okay, norepinephrine. What does that one do?
Norepinephrine is all about alertness, attention, that fight or flight response. Gets you ready for action.
Wow, these tiny molecules sure pack a punch. It's amazing how they can impact our behavior and how we feel in such big ways.
It really is. And some of these adenine neurotransmitters like dopamine and serotonin, they work through these special receptors called G protein coupled receptors.
Those were mentioned in the textbook, but I'm a little fuzzy on what makes them so special.
So unlike those other receptors, which are kind of like fast-acting channels, G protein coupled receptors. They set off this whole cascade of events inside the neuron, like a chain reaction.
So it's less of an immediate thing and more like a slow burn causing these longer lasting changes.
Exactly. It's all about those subtle, sustained changes in the background. And last but not least, we have the peptides. These guys are larger molecules, and they tend to have more specialized functions.
Like endorphins, right? Our brain's natural painkillers.
You got it. Endorphins, they get released during exercise, when we're excited, even in response to pain itself. Then you have oxytocin, sometimes called the love hormone, which is involved in social bonding, trust, all that good stuff.
So we've got our fast-acting amino acids, our versatile amines with their different modes of action, and our specialized peptides. It's like an orchestra with all of these different instruments playing together to create this amazing symphony of brain activity.
I like that, the orchestra analogy. But even with the most talented musicians, sometimes things can go a little off-key. And in such a complex system like the brain, well, sometimes things go wrong.
That's what I was thinking. With all this intricate communication happening constantly, what are the chances that everything always goes smoothly, right?
Exactly. And that's where things can get really interesting, and in some cases, pretty dangerous. The book talked about how drugs and toxins can mess with synaptic transmission, basically hijacking those communication pathways.
It's like cutting the wires or something.
In a way, yeah. Think of something like nerve gas. It completely disrupts that delicate acetylcholine balance we talked about.
Nerve gas. Okay, now that's terrifying. How does that even work?
Nerve gas blocks the enzyme that's supposed to break down acetylcholine, so you get this buildup of it in the synaptic cleft. What happens then? Overstimulation of the muscles, and that can lead to paralysis, even death.
It's crazy how something that disrupts what seems like this basic interaction can have such devastating effects.
It really highlights just how crucial that delicate balance is, and it's not just external stuff that can cause problems. Lots of neurological and psychiatric disorders, they're linked to imbalances in specific neurotransmitter systems.
Well, because if these chemical messengers aren't working properly, things are going to go wrong.
Exactly. That's why understanding these systems, how they work, how they can go wrong, it's so essential for developing treatments for these conditions.
Absolutely. Well, we've gone deep into synaptic transmission today, but it's clear that we've just scratched the surface.
Oh, yeah. We've just opened the door a crack. There's still so many unanswered questions, so much more to explore and discover about how our brains work.
It's mind-boggling, really.
Yeah.
But I hope this deep dive has left you with a sense of awe for the incredible complexity and elegance of the human brain and how these tiny interactions at the synapse level truly shape everything we do. Thanks for joining us today on this deep dive into synaptic secrets. Until next time, keep those synapses firing.